Okay. Let's start with a sound check. Can you hear me in the back row there? Can you hear me in the back row there? Okay, great. Okay, well, we're recording. Before we begin Chapter 19, I'd like to just touch on some general matters having to do with the course itself. Refresh your memory of the schedule. This is in the syllabus, but it may have been a while since you've looked at the syllabus. We have 2 chapters left in the book. So today and Friday, we'll be completing the chapter on transcription and various other aspects of RNA. And if we have a chance, some aspects about the RNA origins of life as an ongoing current area of research. Monday and Wednesday, the 28th and 30th, we'll be doing Chapter 20, the chapter on protein synthesis and modifications and so on. And then a week from Friday, we'll have our last hour exam. Please note that that last hour exam will include all of Chapter 20. This is in contrast to the previous exams where I tried to let there be a lecture in between and so on, but I think that at this point in the course, it really makes things awkward to try to do that kind of thing. I think you can handle it with forewarning. So that the 4th hour exam then will handle Chapters 16 through 20. And in the week of May 5 through 9th, we'll be doing review and integration, where I'll try to kind of refresh your memory of things and pull them together and help you see connections that were there but maybe weren't in the forefront of your awareness because we were so busy with the details. The final exam will be given on May 13th, Tuesday at noon, which is a fairly decent time compared to some, I guess. It is a comprehensive final, so material from anywhere in the course can appear on that. And I will be using some questions from the hour exams on the final. So if you use the previous hour exams to review from and to make sure you're fresh in your memories, some of them will, in fact, turn up on the final exam itself. I don't yet have the rooms to announce for where it is. Do people have any sort of general process questions on either the 4th hour exam or the final exam? Questions? All right, let's move ahead to more interesting things then. So in the last 2 chapters, we've been talking about DNA replication and DNA repair, how precious this core of information is to organisms and how much effort they go through to copy it faithfully, to repair it when something happens. But DNA by itself doesn't do anything. I once shocked a colleague by saying DNA is the dead hand of the past. If all you had was your DNA, you'd be a virus. And so there has to be more to the system for life to go on. And that more that we're going to be looking at is the RNA and protein. And we have to have RNA in our cells in order to have proteins. So the next step that we're going to be looking at is the whole question of RNA metabolism. Your chapter starts off with this rather fetching picture of Amanita phalloides that has the engaging nickname of the death cap mushroom. And the reason for including it in this chapter is that it has among its poisons alpha-amanitin, which is a small peptide that can inhibit the enzyme that synthesizes RNA polymerase. And since if you don't make any more RNA, the RNA you have will go away, protein synthesis will fall off, and over a period of time, you'll stop having new protein. It's an interesting sidelight that what Amanita kills you with is not with alpha-amanitin, but with phalloidin. Phalloidin is a cytoskeletal poison. It polymerizes your actin, and in the process kills your liver. And I can't resist sharing a story about Amanita from my East Coast days. A friend was driving on Cape Cod when he saw an old man by the side of the road collecting what was clearly the Amanita phalloides death cap mushrooms. This guy was a mushroom picker who liked doing it in the wild and so forth. And he felt morally bound to stop and let this old man know what he was doing. And so he asked the man, you gather mushrooms before? He says, all my life. Have you ever gathered this mushroom before? Oh, yeah. Tastes great. What do you mean it tastes great? He says, well, you have to know how to cook it. You slice it thin, and you soak it in cold water. The water will turn black, and you throw that away, and you keep changing the water until it runs clear. Fry it in butter, and it tastes delicious. So apparently both alpha-amanitin and phalloidin are water-soluble. So. Transcription. So transcription is the broad umbrella term we give for making an RNA molecule that is complementary to a DNA template. And so what transcription does is that it's going to copy the information, the sequence information of the DNA molecule into an RNA molecule, which is going to be more active at getting something accomplished in the cytoplasm. It's a hallmark of living things that transcription is selective. I don't know of any cells that simply transcribe everything all the time. The ability to respond differently to different circumstances or during development depends on being able to regulate transcription. And by regulating transcription, we have one of our major ways of regulating gene expression. It's important to remember that a transcript isn't exactly the same thing as a gene. And here we get into trying to uncover how the word developed over time. It is certainly true that the messenger RNA for any protein is a transcript and that it contains the primary sequence for that protein. And so in that case, the mapping of a transcript to a gene product is fairly direct. There are many RNAs, by mass even, in the cell, that are functional but do not need to be translated. It is the RNA that is functional. And the ones that come to mind are things like the ribosomal RNA, which we're going to see when we get to the last chapter, is sort of the connecting core of the ribosome. The transfer RNAs we've mentioned already, which translate the codons into amino acids, and various small RNAs. None of them need to be translated to be functional, and therefore their genes have RNA transcripts but not protein products. It's also commonly the case that transcripts have to be processed in various ways before they're functional, and we'll look at some of that processing. There are regions of DNA that can control transcription, and so they can behave as a gene. That is, if a gene is a region of DNA with a measurable phenotype or a measurable outcome in the organism, these non-transcribed regions that control things have phenotypes. And then on the other side of it, there are some transcripts in our cells, quite a large number of transcripts, that have no detectable effect that we know of. Now, there may be an effect, but we don't know what that is yet. They don't produce proteins. They don't leave the nucleus. If they have an effect, we don't know what that effect is yet. So the relationship between transcript and gene is a little bit less linear than it used to be in the days before we knew so much. But let's look at the human genome. So the human genome is the collection of all of our genes, all of our DNA. Just to give you a feeling for it, each of your cells has enough DNA to stretch from my nose to my thumb, each one of your cells. So you can see there's a lot of room in there for things. And yet it only contains in the order of 30 to 35,000 genes. This was a great surprise when we sequenced the human genome. Going into it, we believed it to be more like 80 to 100,000, but the answer turns out to be smaller than we expected. Only 1% of our DNA has recognizable genes. In us, most of those genes have introns. We're going to see what the relationship of introns is evolutionarily, but in us, most of our genes have introns. There are some parts of the genome that we call open reading frames. An open reading frame is something that has a clear start site for transcription and has internal to the transcript the necessary sequences you need to make it into a protein, but we don't know what it's for. It's an open reading frame. It could be read into a protein, but we don't know what it's for or where it's expressed. We also find pseudogenes, genes that, well, regions of DNA that have homology to authentic genes, but which are now mutated over the years so they no longer can either make transcripts or make proteins. They may simply be damaged. Their transcripts might only be damaged. That is, they might have a start site. They might have a stop site almost immediately. Or they may have lost the machinery or the sequences needed to recruit the machinery to be transcribed. So they're actually been silenced. But you can see from their sequence their homology to authentic functioning genes. And these pseudogenes are one of the tools we have for untangling evolutionary relationships. It turns out that simple genome size is not the best clue to the complexity of the organism, partly for this reason, that the whole of the genome is not necessarily taken up with genes. But a better guess at it is from looking at the number of regulatory genes and regulatory regions. This is just giving you a little taste of part of the human genome. It's showing you known genes, new genes, meaning open reading frames that we're still in the process of figuring out, pseudogenes, things that have been damaged one way or another, and putative sequences of one kind or another. And you can see that there's really a lot of space in between for the known genes for a lot of other things to be happening. So when we're talking about macromolecular metabolism, DNA, RNA, protein synthesis, for all of these processes, there's something that gets it going, there's a way to continue doing it, and a way to stop. Those are general features that have to be satisfied for these things. So to start off with, we will look at the initiation of transcription and how it's organized. Now, interestingly, transcription is like replication in that it is organized in the nucleus in very discrete domains, in what have been called factories. And I think Lynn may have shown you this picture of the localized replication in nuclei, so that DNA replication doesn't happen simply anywhere. It happens in specialized domains. Here is a way, these two same nuclei, but now it has been stained in such a way that it only highlights the transcriptional factories. And because you can compare these, you can look over here and see that there's almost no overlap between the red and the green, which means that the specialized sites are either specialized for replication or they're specialized for transcription, but they don't do both. That wasn't obvious in advance. Both of them involve treating the DNA as a template. But it turns out to be the case that replication happens in one place and transcription happens in another place. One of the most important jobs of the RNA polymerase is to be able to keep the growing RNA chain from getting tangled up in the DNA. And so if we take a look at a sort of a very macro view of RNA polymerase, DNA is fed into it. It has to open up somehow or other so that you can get access to the sequence. Part of it is transcribed, and then that growing transcript is diverted away from that site so that it doesn't simply stay with the DNA or get tangled into it or whatever. And then as it exits, the DNA is allowed to reanneal. So beginning transcription is an extremely important decision for cells, since what you transcribe is what you're going to have to work with in the next minutes to hours. Very important to pick the right things to transcribe. In prokaryotes, let me back. The word promoter is used for the sequences of DNA by which the machinery knows this is where to start transcription. So promoters are the street sign that says start here. In prokaryotes, there are two consensus sequences that are generally found at the beginnings of genes that are transcribed. One of them is found at -10, meaning 10 nucleotides from the first nucleotide in the transcript. And it's called a TATA box, sometimes TAT, AAT, so you can see why it'd be called TATA box. And then further upstream to the 5' side of the start site is another consensus, TTGACA, at the -35 region. In order to identify genes and select which ones to work on. The E. coli polymerase has several subunits. It's an alpha 2 beta beta prime tetramer with a sigma subunit. And the sigma protein is a protein that contributes the specificity for which of the promoters this particular molecule of polymerase will be used. And so there are different sigma compounds or different sigma proteins for different classes of promoters. In eukaryotes, transcription also begins at promoters, but promoters are a little bit more complicated in eukaryotes than in prokaryotes. Again, the start site is again called the +1. In eukaryotes, there is a TATA box, TATA, goes on a little longer, at about -24, so it's further upstream than the TATA box in the prokaryotes. But the other upstream sequences are immensely varied, a huge variety of possible consensus sequences and structures and so forth. And those are found in the 5' domain further upstream from the start site. To give you a sense of just how cosmic and Byzantine eukaryotic promoters can get, there are functional promoter sequences as much as 3 and 5 kilobases, 1,000 bases, upstream of the start site of transcription. So it's not at all uncommon for there to be a gene in which there's more regulatory regions and more untranslated regions than there is actually coding regions for protein. An awful lot of the sequence information of a eukaryotic gene is involved in kind of the bookkeeping and regulation. Eukaryotic polymerases do not have sigma factors, but they do have a lot of transcription factors, and we'll look at some of those. These are factors that are both protein-protein and protein-DNA interactions to ensure that the correct things are transcribed at the correct time. It's actually kind of amazing that we can find any of the eukaryotic promoters. You remember I said that maybe 1% of the human genome was actually genes, and the promoters, the actual specific sequences that trigger transcription, are fairly small. So it's a very small fraction of the DNA that is the promoter. And then to increase the difficulty of finding a promoter, as you remember from Lynn's talk, DNA isn't naked. DNA is always coated with histones and packed into chromatin, which further complicates the search for trying to find those particular sequences for promoters. Heterochromatin is very condensed, so that the accessory proteins in heterochromatin lead to it being quite compact, even during interphase. And it is commonly found that that heterochromatin only has a few genes in it that are transcribed. Euchromatin is the word we give for the more open part of the DNA. It's not as tightly packed. Many of the histones in euchromatin are acetylated. Here's an example of how an acetyl group on a lysine would look. And you can see that acetylation changes the charge on that lysine so that you don't have the positive charge there. It lets it come peel off a little bit from the negatively charged phosphate backbone of the DNA. And there's a growing sense that there's some kind of a histone code or symphony that contributes to how specific transcription factors are selected to interact with a part of the DNA, so that at rest, so to speak, this sequence of DNA has bound on it histones and other accessory proteins, and this region of it may be made more accessible by being acetylated, and which will then recruit factors, and others less accessible by having that removed. Now we can talk a bit about what are called general transcription factors. The general transcription factors are the transcription factors that are basically needed by genes in general. These are not the ones that make for great specificity. There are additional factors, which we don't have time to go into here, that confer tissue specificity, age specificity, metabolic specificity, and so on. Polymerase II is RNA polymerase II, and it is responsible for, I'm so sorry. Sorry. It is responsible for making messenger RNA. And we name the transcription factors brilliantly by the name TF for transcription factors, and then with a Roman numeral for which polymerase. So anything called TF2 is a transcription factor for polymerase II. And then we get on with creative names like A, B, D, E, F, and H. One of these in particular is especially striking. TF2D is a transcription factor that plays a pivotal role in a lot of this. Its name before this nomenclature was developed was TATA binding protein. Now you remember there's the TATA sequence at about -24. The TATA binding protein is something that can bind to the TATA sequence, which you might well imagine is an important part of getting transcription started. TF2D, or TATA binding protein, can interact with TAFs, which is TATA binding protein associated factors. And so TF2D is sort of a complex of these different things. TF2D, or TATA binding protein, can bind to the TATA sequence and actually bend it a little bit there. And then the additional TAFs, the TATA binding associated factors, TATA binding protein associated factors, are different in different tissues. And they are part of why some genes are transcribed and others are not. They're not the only thing, but they are one of the contributing factors. They often have sequence-specific interactions with the DNA. Some people have called TF250 the Swiss Army knife protein. It has at least 3 activities, enzymatic activities of a kinase activity, which you can imagine by phosphorylating TAFs can interact with them to make them more or less higher affinity. Histone acetyltransferase, and we've talked about how acetylation tends to make the histones loosen up and make the DNA more active, and a ubiquitinylation of H1 for proteolysis. Now, this is looking ahead a little bit. Ubiquitin is a protein, some very small protein that can be added to other proteins to make them degraded. And so if you were to ubiquitinylate H1, histone 1, you would tend to make it go away, and that again would help to open up the chromosome structure. There are regions of this and other proteins called bromodomains. And if we look at the structure of the protein interacting with the targets on the other proteins, there are acetyllysines from histones that can interact into that little pocket there. And these bromodomains are found on a number of proteins that interact with the histones. So we start by building things up. We have to have TATA binding protein come in and bind. In this diagram, you can see how the TATA binding protein has really kind of kinked the DNA in the process of binding to it and starting to open it up. And we've added on TF2A and TF2B that have now bound to TF2D, or TATA binding protein. TF2H has a helicase activity. And a helicase activity would be very useful for opening up the DNA helix to make accessible that template sequence that we need to make a complement to. And then when this protein complex there is sufficiently complete and complex on the DNA, it acts as a landing pad or a dock to which polymerase II can land and interact. And when the whole thing is assembled, we talk about a transcription bubble because there's a region of the DNA that has to be opened up for the whole thing to go further along. Now, there are additional layers of development and control here, some of which are surprising, if not actually a little bit spooky. And a couple of these are called enhancers and silencers. These are the names given to some of the regions that can influence transcription from a distance. So that the TATA binding protein is going to bind at the TATA sequence, which is very close to the startup transcription. But you can have sequences in the DNA that are quite a long way from the promoter that nonetheless change the activity of that promoter. So in very, very generic terms, if you have a promoter here and an enhancer at some distance, this enhancer may change the activity at that promoter by many orders of magnitude. A silencer would be something at a distance which can reduce activity many orders of magnitude. So that between the two of these, we can get up to a 10 to the 9th fold range in how much a gene is transcribed or not. Now, one of the models for how enhancers might do this looks something like this. Here's just the naked bare DNA pulled out straight. Enhancers usually depend on some kind of a protein interacting with them to accomplish this. So here's some activator there. Here you have a promoter that has the general transcription factors we've been talking about. It has an RNA polymerase. And that this DNA then loops around, and that brings closer to the promoter both the activator and in some cases some kind of a connecting protein that brings the things together. One enhancer may turn on multiple genes, so it's capable of changing the activity of more than one gene. Okay. We talked a very little bit in the integration of metabolism about the idea of hormones and that hormones can integrate metabolic behavior. We talked a little bit about how some of those things integrated at the level of the cell and the level of enzyme activities. But hormones can also act by actually changing gene expression. And to look at some of those, so steroid hormones, those would be things like estrogen, testosterone, cortisol, the mineralocorticoids, like the ones that are missing in Addison's disease, thyroxine, retinoic acid, which is a relative of vitamin A. All of these can diffuse through the membrane. They don't depend on a surface transmembrane receptor the way insulin, for example, does. And having gone through the membrane, they can bind to a receptor inside the cell, and when they occupy that receptor, and only when that receptor is occupied, it then binds to the promoter and changes activity. It's interesting to note that at least for some of these things, the receptor is present outside of the nucleus, and when it's occupied by the hormone, it moves into the nucleus and binds to the appropriate sequences. Even for those hormones that do act through transmembrane protein receptors and have some direct metabolic influence right away, some of those have additional levels of control at the level of transcription through STAT proteins. And STAT is an acronym for Signal Transducer and Activator of Transcription. And so one of the consequences of a signaling cascade, whether it's mediated through cyclic AMP or through calcium and phosphoinositol, PIP2 or something like that, they can activate these STAT proteins which need to dimerize, and when they dimerize can then bind to and bend the DNA molecule in ways that make it more accessible. In some cases, you'll need several things at once, and so it might be useful to be able to coordinate transcription. This is especially obvious in prokaryotes, who have a nifty way of doing these things. In prokaryotes, we find a number of key genes in what are called operons, and an operon is a cluster of genes, several in a row, which are all related somehow. related in their function, related in their substrates, whatever. And then these genes can be controlled as a unit, being turned on or being turned off as a unit. And whether it's controlled by turning things on or by turning them off depends on what you're looking at. So, for example, tryptophan, TRP is, again, tryptophan, right? There's a tryptophan operon. You may remember we didn't go through the exact sequence, synthesis of tryptophan because it's complicated. That's why it's one of our essential amino acids. There's a lot of genes in the tryptophan operon. And the E. coli is stingy. They don't want to waste time making their own tryptophan. And so the tryptophan operon is turned off if the concentration of tryptophan is high. And it's turned on when the concentration of tryptophan is low. And this is a schematic of that tryptophan operon. Here's the P for promoter. And here are the various genes in the tryptophan operon. Prokaryotes can pull a trick that we ourselves don't, which is when after the promoter is activated, when the promoter is active, the transcription can go clear through multiple genes. And so a single RNA transcript would encode for TRPL, E, D, C, B, and A, would go all the way through. And then that one messenger RNA would have multiple places for translation to start at it. So you'd have like a Christmas tree coming off of that one messenger RNA. One of the products of the TRP operon is a repressor. And that repressor can interact with tryptophan. So when tryptophan is present, it binds to the receptor. That makes the receptor active now, and it binds to the promoter and turns off transcription. If the concentration of tryptophan falls low enough, that repressor will at some point fall off accidentally and not come back on because there's no tryptophan around to get it back on. Another operon that has taught us a great deal is the lac operon. And the lac operon gets its name because it is the set of genes that are involved with lactose metabolism. You remember lactose is a disaccharide and present in milk. And for lac operon, in the lac operon, there are fewer genes than for tryptophan. There's a beta-galactosidase that can split the lactose into two monosaccharides. There's a permease, which, as you can guess, is involved with letting the membrane of the cell be permeable to lactose. The assumption is if there's more outside than in, it'll just run downhill. And LAC-A, which is a transacetylase, it was a mystery for many years why it was in that operon. We now think it may be protection from non-hydrolyzable sugars that might contribute to aging. There's a gene I that makes a repressor. This protein binds at the O operator site, and it blocks the movement of the polymerase in the absence of an inducer. Now, the inducer is not actually lactose, it's allolactose, which is a slightly changed version of lactose. And so what that looks like is something like this. Actually, it kind of looks like Bozo the Clown. I don't always know why protein people pick the colors they pick. So the lac repressor, shown over here by itself, is a dimer of dimers. So it's a pair of two proteins in each one. And it acts like a pincher to grab to the DNA. And this is what the lac operon looks like when the lac repressor has grabbed onto it. And you can see how it's grabbed that DNA and holds it in a constrained configuration, almost looking a little bit like a nucleosome or something, inside-out nucleosome. And the polymerase can be bound at the promoter site, but since it's looped up like this, it's not going to go anywhere. And what this means is it's like a spring-loaded operon. If the inducer comes in, this lac repressor will drop off, and the gene is ready and loaded, and transcription will start immediately. So it allows for a very rapid response when circumstances change. It's interesting to compare the structural strategies of some of the common DNA binding proteins. And all the proteins on this page are DNA binding proteins that are involved with regulation of one kind or another. So for example, this first one here is the lac repressor, which is one example of a helix-turn-helix family who have a similar strategy for how they bind to DNA. And it fits here into the major groove of the DNA. Here's the minor groove there. Leucine zippers, you may remember back from when we were talking about protein structure. They're an example of a coiled coil. Leucine zippers may be either two of a kind or two of different ones that contribute to more complexity. And they act like kind of a pincher also, grabbing onto the major groove. Again, you can see the major groove. And we also talked about zinc fingers when we talked about protein stability. They're very common in transcription factors. And here's an example of one zinc finger fitting into the major groove again. So there's kind of a theme there. So let's talk a little bit about the enzyme itself, about RNA polymerase. In eukaryotes, we have multiple RNA polymerases. We had multiple DNA polymerases. We had a bulk one, and then we had a variety for other special purposes. In the case of the RNA polymerases, there are at least three major ones that do quite a bit, each of them, and then some others as well. Polymerase 1 is responsible for the ribosomal RNAs. So its only target are the ribosomal RNA genes. Polymerase 2 has as its target the messenger RNAs, so it is responsible for producing all the transcripts that will end up making a protein. Polymerase 3 makes tRNAs and a variety of other small RNAs. One way of remembering the sequence here is it goes from the biggest RNAs to the smallest RNAs in general. That is, in general, the initial transcript of the ribosomal RNAs would be bigger. In addition to these three polymerases, 1, 2, and 3, that are found in the nucleus, eukaryotic cells also have organellar polymerases. These are RNA polymerases found in mitochondria or in chloroplasts. Since there are genes in there, there are certain components of both of those organelles that are encoded by the organelle DNA. And so those polymerases are essential for life. Interestingly, those polymerases sometimes have components that nonetheless are coded for in the nucleus. There is a common structural strategy for making an RNA polymerase. If you think chemically of what RNA looks like and what DNA looks like, they're really very similar chemically. And so it shouldn't be too surprising that an RNA polymerase has a palm, thumb, and fingers comparable to the kind of structure that Lynn talked about when she was talking about the DNA polymerases. Metabolically, catalytically, it also uses magnesium, as the DNA polymerases do. Like DNA polymerases, all of these polymerases synthesize chains 5' to 3'. These RNA polymerases, to be a true RNA polymerase, will need a template. There's some kind of a template molecule that they are copying from. But unlike DNA polymerases, they do not need primers. RNA polymerases can start in the middle of the nowhere and get going and make a molecule. You may remember when we were talking about DNA structure that there are three major forms of DNA found, A, B, and Z DNA. B DNA could be thought of as bulk DNA, if you like. And we talked a little bit about A DNA being squatter and wider than B DNA. If you think about it, while you're making a new RNA molecule, there will be at least a little stretch of that that is an RNA-DNA hybrid. And so there is a little stretch in that new transcript that is in a DNA-RNA hybrid form. And the overall features of a DNA-RNA hybrid are most similar to the aDNA form of the straight DNA. DNA enters the polymerase and gets separated so that the two strands are separated from each other. And when they get to the polymerization site, the DNA has to kind of elbow out of the way to make enough room for the nucleotide triphosphates to enter. And the sequence in which nucleotide triphosphates enter the polymerase is random. There's no selectivity at that point in the process. It just keep trying one until one is the right one. Another feature of RNA polymerase is that it is highly processive. We talked about this before in terms of DNA polymerases. We talked about a little bit about some enzyme, a few of the metabolic enzymes, the ones that process glycogen, for instance. But certainly, these are far more processive than those. And that makes sense because the RNA polymerase has to get on at the promoter and stay on and stay on and stay on until the whole thing is made. So you want to have an enzyme that is really very processive. And part of that is accomplished by there actually being a kind of a lowering of a part of the polymerase, like a clamp, that can tighten against the DNA and keep it in there so it can't get out of there. The polymerase will only use single-stranded DNA as a template. So if the DNA had not separated entering into the polymerase, it would not be usable as a template. And you can see that it must be pretty processive when you take into account the fact that genes can be anywhere from 1,000 to 1,000,000 nucleotides long, or over 1,000,000 nucleotides, actually. So if we want to kind of get a big picture of what's going on here, here's sort of a cutaway view of the inside of a polymerase molecule. We have DNA entering from the downstream side. The helicase activity is opening up the DNA so that we have naked strands for it to work with. This sort of region of the polymerase acts like a funnel that allows nucleotide triphosphates to enter at that point. And the decision is made in there about whether or not it's complementary or not. As that transcript is made, it leaves the polymerase that way, and the DNA that's been transcribed reanneals to be a double helix again. But you can see that it's very much structured to make sure that there is a naked template available, but that it's naked for no longer than it has to be. Now, we've talked about how DNA replication involves some proofreading, that the DNA polymerase can correct some errors. We're going to see in protein synthesis that there is some proofreading in that process. And it has been recently found out that RNA polymerase can also proofread. And the way that works is something like this, that if a incorrect nucleotide not merely drifts in through the funnel, but actually gets covalently attached to the growing chain, that's going to be a mismatch at that one spot, and it will distort the helix. And that is a signal to the RNA polymerase to basically backtrack on the RNA and trim off that incorrect base so that you can actually back out that entry point and have that incorrect one eaten off. So far, we've been mostly focusing on initiation and the actual mechanism of adding another base. But there's this long phase in between, elongation, that has to happen for there to be a long enough transcript to be worthwhile. And we mentioned that they're processive. But what's interesting is when we look at transcription in detail, what we find is that many short RNA transcripts are made and then discarded. And that of the times that the RNA polymerase lands, it's only occasionally that it somehow undergoes a transition to this locked-on, highly processive, let's keep at it, chugging along kind of machinery. So what's the difference? Why is it generally aborted and occasionally functional? If we look at an initiation complex like this, we have the promoter sequence of the DNA. We have here some general transcription factors like TF2D and so on. We have special factors for that particular promoter. And here's your RNA polymerase. It has a tail sort of sticking up here like a teapot spout or something. This is called the CTD, which is short for C-terminal domain. So this is one end of that protein. It's the C-terminus. And the C-terminal domain has a very peculiar sequence. Tyrosine, serine, something, proline, tyrosine, serine, something, proline, serine, 52 times. 52 copies, so this very peculiar looking sequence. And it's just sort of sticking out there. It's not going to, I mean, with all those prolines in there, it's not going to fold very nicely. It's kind of flapping out there in the breeze. And these serines are potentially targets for being phosphorylated. So a kinase can phosphorylate those serines and modify them. Now, if you think about this long sequence, multiply phosphorylated, that's a lot of negative charges in there, a lot of acid charges there. Sometimes this domain has been called the negative noodle, which may be a good way to think about it, that once it's phosphorylated, it's a negative noodle. And that change in charge on it allows it to release the mediator, this M complex that's somewhat specific to this particular promoter, and pick up additional elongation factors and charge on down in a very processive way. The general transcription factors are left behind. They stay on at the promoter. And so this means that once you have effectively initiated at that promoter and moved on, it is now a better place for another promoter, another polymerase to come in and make an RNA transcript. This is one of the things that makes transcription at an active gene more productive than an inactive one. So what about the histones? I mean, this previous picture, this picture makes it look like DNA is just as straight as a pencil, which of course we know it isn't. DNA is packed onto nucleosomes, onto beads on a string. So how does the RNA polymerase deal with the fact that DNA in the nucleus is bound as beads on a string around histones? One thought is that the histone acetyltransferases are loosening those associations with the DNA and make that easier to access the DNA. It is clear that even with the activity of histone acetyltransferases, the DNA does not become stripped naked. And this is believed to be some of the ways in which an RNA polymerase can, in effect, read through a nucleosome. So you can see it coming in and approaching the nucleosome. As it opens up a loop, it can be reading off this part of the DNA while that part of the DNA keeps its toes down on the histone core. The RNA polymerase continues on and comes out the other side. So the DNA has never been completely bare, even though the polymerase has passed over it. I think we'll call that it for today.